is no additive or synergistic effects, not even an additive effect. So it means probably they don't work using the same effects. We know that amifostine is a free radical scavenger, so which takes the free radicals out of the solution, and probably the deuterium depleted water functions using completely other ways. So, compared, however, to the administration of hydrochloric ambihine, we have observed that the test group here above has a significantly increased survival compared to the control group. So it means that even the chemical radiomimetic was, so the mice were protected against the chemical radiomimetic. So we see here the effects, and we were able to calculate also the dose modification factor. The DMF, the dose modification factor, is a numeric quantification of the capability of reducing the dose. The DMF was 1.41. Okay, so administered together with a known chemical radioprotector, it does not significantly increase survival. The lack of any synergistic or additive effect between the deuterium depleted water and amifostine suggests that their action mechanisms are not different, or that radiation protection has a maximum which cannot be overcome. It's also an idea which is not mine, it is of the greatest professor in the Walter Reed Army Institute, that say at a certain point, never mind how much biological screening we do, we cannot protect the animal or human organism against ionizing radiation completely. So we were not able to save it. The calculation of the dose modification factor is an average DMF of 1.41. This is not huge compared, for example, with the DMF of amifostine, 1.81. But we have to know that amifostine is a substance which is highly toxic, and it has to be administered absolutely in the first minutes after the irradiation in order to have some effects. And we cannot give it for a very long time because as a sulfhydrylic derivative is highly vasodilator and hepatotoxic and so on and so on. So it will protect us against the some effects of radiation, but then it will kill us by destroying the liver. So I'll take anyway this way because it's not so toxic. So what have we seen? We looked at the intestinal mucosa to see in the control group we have observed what we call necrotizing enteritis with cytolysis and loss of microvilli. So the surface, the internal lining of the intestine, of the small intestine, is completely slowed off. So it means that the cells have been destroyed here, and they are going to be eliminated, and they will leave holes in the mucosa of the intestine, which will produce bleeding diarrhea. It will leave openings into the intestine for the microbes to get inside, and on the, let's say, on the background of immune depression produced by the radiation will produce septicemia, and the animal will die. However, when we look in the deuterium depleted water treated group that received 7.5 grays of gamma radiation, we see that there are some mucus ulceration, but it keeps the microvilli and the amount of necrosis is less. So it means some way, somehow, the small bowel has been protected by this ingestion in a way or another. We looked at also at the liver. So what have we observed? In the group which has received normal water, we see the loss of normal architectures, and we see areas of necrosis and apoptosis. This does not look like a normal liver at all. We see here shrinking cells, yeah, signs of apoptosis. Yes, we don't see the lobuli, which have to look normally. And we see that in the group B, it will keep the normal architecture and it won't keep the necrosis. We see the centrolobular capillaries here. So also the hematogenic bone marrow. So we cut a little the bones and we made slides out of them, and what have we seen? We have seen the loss of cellularity. Excuse me. we've seen the loss of cellularity, so it means it loses cells. These are bone marrow blood cell precursors, which were kept. We tried to test the antioxidant mechanisms. We have observed, so I don't have the time to continue this, but we have seen reduced and statistically significant increase of superoxide dismutase activity in liver and no effect in brain. Malonyl-dialdehyde, which is a marker for peroxide, for lipid peroxidation, is significantly lower than brain and no changes in liver. Insignificantly reduced glutathione and total thiol groups, which seems that overall deuterium-depleted water does not seem to have antioxidant effects. So probably it does not work this way. So let's see in the immunologic screening. At the first level, we have tested the hematologic aspect by counting the RBCs and WBCs, by checking leukocyte formula. Then we also tested the T lymphocytes counting with the B lymphocyte countings and also the phagocytosis analysis, the peripheral blood neutrophil phagocytosis assay, peritoneal macrophages and serum complement. The testing is not so difficult. In fact, it's just taking some quantified samples of microbes and then they put it in contact with serum from the animals, and then you seed them on another tube. And if they have been destroyed, they will grow slower. If they haven't been destroyed, they will not. They will grow in a normal way. So what have we observed? Oh, sorry, excuse me. That the phagocytosis in the peripheral blood is increased. ASD is Romanian for deuterium-depleted water. Serum complement activity is also increased, yeah? And also the phagocytic capacity is increased by seeing the remaining colony-forming units of bacteria that we have used as controls. So the remaining is much less reduced. It means they have been killed by the macrophages, yes, the macrophages from the peritoneum, which is a very good thing, which means that the organism is able to defend itself against various types of infections. So also the group receiving 2 weeks presented significantly higher leukocyte count than the control group. The absolute values are not high enough to suggest leukocytosis, so they're not high enough to suggest that it will have an effect similar to an inflammation. The feeding with the deuterium-depleted water for a couple of weeks did not significantly influence the number of T lymphocytes. The number and activity of B lymphocytes was a little increased, but not significantly thus. However, it stimulated the phagocytic capacity of polymorphonuclears in the peripheral blood and of the peritoneal macrophages. So they are all significantly increased. This is the effect that I've been telling you about. Now we tried some standard cell cultures. Unlike that, the results that we have observed earlier, what have we observed? So we tried 3 types of cells that we had available at the time. So it was mouse renal adenocarcinoma, mammary adenocarcinoma, also from mouse, and normal fibroblasts. So what have we done? We have used the completely normal cell culturing techniques, with the difference that the RPMI medium that we used was reconstituted. It was powdered media, and then we reconstituted with various concentrations of deuterium-depleted water. And what have we observed? We have observed that for the first 3-4 days, the cell count and the population doubling time and the population doubling levels have increased significantly. Now look, how do the cells grow, yes, with 90 parts per minute per million compared to the normal. So they grow both neoplastic tissues, so adenocarcinoma, renal adenocarcinoma and mammary adenocarcinoma, and the fibroblast with a completely different configuration of the curve, however. So for example, how does it look? Yes, after 48 hours, it's almost confluent after 48 hours, while we have only the normal doubling in normal water, that's normal fibroblast.